Hello everybody, this is Dr. Eduardo Beltran. Welcome to this module on the microbiota. Today we will be talking about an axis that I describe as the Microbiota-Nutrient Metabolism and Epigenetics axis, which is actually very important because, believe it or not, your microbiota on a daily basis is telling what your genes are supposed to be doing. We don't think about it very much. We don't sit down and think about that the bugs that we have in our microbiota are actually telling us what genes to be turned on and turned off. And today we're going to go ahead and dive into this fascinating field of the microbiome and see how it has a direct impact on nutrient metabolism and also on the way how certain genes are methylated and also some certain genes are turned off through acetylation or deacetylation. And we will also have an idea of how certain things in regards to our epigenetic expression in regards to vitamin D and how vitamin D regulates our epigenetic expression as well. So let's go ahead and begin. And once again, I'd like to go ahead and welcome everybody that's joining us today. And let's go ahead and dive in. So the microbiota, as we know it, has many different functions. One of them is it participates actively in the digestion of fiber. And as we know, specific bacteria are responsible for being able to ferment and metabolize fiber and transform them into small chain fatty acids such as propionate, butyrate, and acetate. Then we also know that the microbiota has a very big impact on how we make our neurotransmitters. And a good example here would be, for instance, serotonin. We know that approximately 90% of all the serotonin that's made in the body comes from the gastrointestinal tract, and only 10% is actually made in the central nervous system. So that's something very considerable. And we also know that these bacteria are also very important because they also help out not only in the making of serotonin, but down the road participate in the making of melatonin, and also other neurotransmitters such as dopamine and acetylcholine as well, and norepinephrine. So neurotransmitters are very important, and our microbiota has a very big impact on how we make them. Our microbiota also makes many different hormones. Okay, that's a different video to go ahead and discuss how those hormones are made, but I'm just going to go ahead and just briefly mention them. And we also know that the microbiota participates actively in the making of many different B vitamins, such as folate, B12, okay? And we'll see the implications that this has in how the methylation cycle takes place. But it's very important to recognize that, yes, bacteria that we have in our gut, they do make certain vitamins, such as vitamin K2, vitamin K, vitamin B9, vitamin B12, and many other vitamins that are very important to be able to help out in the epigenetic expression and in the making of methyl groups, and also in the making of certain small chain fatty acids that are essential for upregulating and downregulating certain genes. So let's go ahead and look at how that also happens. But let us not forget that 80% of our immune system is found in the gastrointestinal tract, and the microbiota has a constant interaction with our immune system, which basically is always educating our immune system to be able to recognize what is a bad pathogen versus a good pathogen, right? And teaching us, in other words, how to be able to develop an adequate immune tolerance. Okay, so the microbiota participates actively in educating the immune system in how to behave, right? Now, let us not forget that the microbiota has a barrier that separates it from the immune system, known as the mucin layer, and these small chain fatty acids are very important in order to be able to help make such a mucin layer, and also in the renewal and the recycling of the mucin layer. There is a video that I already made, and that you can go ahead and check that out if you'd like. I highly suggest it. I mean, that would give you a good basis in order to be able to understand the importance of how small chain fatty acids participate in the renewal and also the recycling, the making and the recycling of the mucus layer that separates the microbiome from interacting with your immune system. But in general terms, let me just go ahead and summarize. The microbiota is very important in the digestion of fiber, the making of neurotransmitters, the making of hormones and vitamins, and finally educating our immune system. Okay? Now, in the case, for instance, if we have an example here of a patient who suffers from obesity, and let's say that he also has he has diabetes type 2. Well, if we look at the microbiome of this individual, we're going to be noticing that there's going to be a shift in regards to diversity. There's probably going to be a predominance of certain bad bacteria over good bacteria, right? And as we know, whenever we have dysbiosis, which usually is the case in individuals who do have diabetes or obesity or both, that's a type of form of dysbiosis that promotes inflammation. And one of the ways how we can actually look at this is not only just by looking at the microbiome itself, but also we could look at other metabolic markers which are going to be altered, such as an A1C that might be a little bit elevated, or perhaps fasting insulin might be elevated. And if we even look even more closer to the GI permeability, like markers such as zonulin, that might be actually elevated. All of these things are suggestive that this individual has dysbiosis, and which can later down the road end up causing any form of disease process, such as autoimmunity or even cancer, depending on what pathogen is causing the whole shift in regards to the microbiome. Now, a study that was done back in 2016 and 2017, thanks to Kramer, Alexandrova, and also Romano, they were able to demonstrate that the microbial colonization regulates the global histone, acetylation, and also methylation processes that are found in multiple host tissues. And this happens thanks to diet-dependent factors or manners. Now, let's go ahead and recall a little bit of how our DNA structure is. As you guys know, here we have a double helix of our DNA. Over here we have our bases, nitrogenous bases, which can be purines and pyridines, adenine, thymine, cytosine, guanine, and so forth, right? So here we have our base, right? Our nitrogenous base, which is bound to a pentose sugar, such as D-ribose in this case. That's why I like giving patients D-ribose, because we're allowing to be able to help in the making of a nucleotide. And this D-ribose, or this pentose sugar, is bound to a phosphate. So the presence of a phosphate, a pentose sugar, and a base, a nitrogenous base, makes up what we know as a nucleotide, correct? And the combination of two different nucleotides will end up making a combination of different bases, which when they pair up in a specific group manner, they might actually be coding for a specific gene. Now, once this double helix, as you can see over here, gets wounded up around what we know as histone, these are known histone proteins. These histones are represented usually by lysine and arginine, okay? And when they start getting wound up around over here, these structures, these histones are very important, because if they get acetylated or methylated, they can actually be more compact, or they can be loose, set out loose, right? And we know for a fact, for instance, if we look at our DNA structure, here we have our double helix DNA, which gets bound into these histones, which form a nucleosome. And the more they get compacted all together, they end up making what we know as a chromatin fiber. And this chromatin fiber gets flat out, set up on, it lies on itself. And as you can see, they make a huge structure known as a chromosome. So here we can see how our DNA is basically set up to be, right? All right. So now, if we look at the whole process of DNA methylation versus histone acetylation, we have to always remember that our DNA is made by nucleotides, and nucleotides have a base and a sugar and a phosphate. And it's also bound to another base. Let's say in the case of guanine, will correspond to cytosine, right? And there's a hydrogen bond right here, correct? So in this case, let's say in regards to DNA methylation versus DNA demethylation, in this genetic sequence that we see here in our DNA, right, we see that there are no methyl groups that have been added. But if we add a methyl group to cytosine in this case, for instance, right? For instance, here we have cytosine, right, which is a nitrogenous base, and thanks to an enzyme, which is an enzyme methyltransferase, it's going to add that methyl group to that nitrogenous base, converting the cytosine into a methylated cytosine known as 5-methylcytosine, as you can see right here, you see? So it's important to understand that the methylation process requires specific enzymes that do that function for us, okay? And then once that this gene or the sequence has been methylated, this whole area now perhaps might be, turned off or turned on, okay? Now, the methylation, usually what it does is that if there are methyl groups, these methyl groups, they're going to keep transcription from occurring. So in other words, transcription is going to be blocked. In other words, it stops the protein synthesis, okay? Now, here we have the DNA methyltransferases that I just mentioned over here. You see that? All right. Now, if we do a demethylation, let's say the taking off of these methyl groups, right? The removal of methyl groups, this now allows for the DNA to be transcribed, okay? So transcription now is unblocked, and proteins and enzymes can be upregulated and made. And here we have an enzyme known as histone demethylases that allow this to happen, okay? All right? So now, in the case, for instance, if it's a normal cell, you can see that there are methyl groups right over here. But when we look at a cancer cell, we sometimes are going to be able to see that there's a lot of different methyl groups that are added upon many genes. And sometimes some specific oncogenes, such as P53, which is a gene that's responsible for DNA repair, it might be methylated. And the reason why is that's because it's actually inhibiting the repair of that cell. So this is one of the characteristics that we sometimes see in cancer cells. Now, in regards to histone acetylation versus histone deacetylation, here we can see acetylation taking place, right? Thanks to an enzyme known as histone acetyltransferases, where there's an acetyl group being added to these histones. And by doing this, transcription now is unblocked. You can actually see that these histones are kind of separated, and that kind of makes it that DNA kind of loosened out now, and also allows for transcription to take place. Now, in the case, for instance, when we have histone deacetylases, what happens is now that there's a removal of these acetyl groups, and now these histones are tightly packed together, and it doesn't allow for transcription to happen. So the deacetylation process blocks transcription, and thanks to these enzymes known as histone deacetylases. So histone deacetylases block transcription, and histone acetyltransferases promote transcription. Okay, and what's the major difference? That when we have transcription, these histones are going to be loosely found, and when there is deacetylation, they kind of bind back together and get more compacted, which does not allow for transcription to take place. Now, here's another image. As you can see, here we have deacetylation taking place, right? The removal of acetyl groups, and here we have acetylation taking place. So here you can see it's nicely compacted together, right? And here we're having the addition of acetyl groups. Histone acetyltransferases and histone deacetylases. Now let's go back to a very interesting study that was done back in early 2000s, right? At the Duke Medical Center, where some scientists were doing research on agouti mice. Here we have two agouti mice, and believe it or not, these are two sisters that are genetically identical. They're exactly the same. They say they have the same genetic makeup, but as you can see, phenotypically, there's a very big difference between one versus the other. And the main reason here is because one of these, this agouti mouse over here, received a healthy diet that was rich in methyl groups and many vitamins and minerals, and versus this other agouti mouse that just basically received normal mouse chow. Okay, and as you can see, the more of the methylation that happened, the more the phenotype was preserved. But when methylation did not take place, one of the things that we started to notice that the agouti gene was expressed, which caused a series of certain phenotypical manifestations to show up, such as obesity, the change in hair color, right, and prone to other different diseases when compared to the one that was mainly more methylated. Okay, all right. Now, interestingly, is that the offspring of these agouti mice that received a diet that was rich in methyl group, we were able to see something very interesting happen here. Basically, 75% of the offspring preserved a healthy trait, which were lean, dark brown, and were not prone to obesity, right? In regards to the one that already was on a normal diet, 75% of that offspring was similar to the mother, and 25% was different. So basically, in other words, what I'd like to go ahead and say here is that whatever the mother eats during pregnancy, okay, and the way how her epigenetics is being expressed will also be passed down to her offspring. So this has very huge implications. All right. And as you can see in this picture over here, we have two adult women that are genetically identical, twins. All right. These are 34-year-old twins and could not look more different. Mary was 420 pounds. Sorry, this is Mary. And Ruth, her sister, was only 120 pounds. So what happened? What happened here is what happened is that the microbiome changed, and the dietary habits that we have throughout life, start changing and making a huge, humongous shift that changes the diversity of the microbiome, which ends up causing epigenetic changes in our body. That's right. So here, the reason why one individual is different from the other is because the dietary habits change our microbiome or microbiota diversity, which also is going to have a big impact on how our epigenetic expression is going to take place. Okay, so changes in the diet modifies your microbiome, and this modified microbiome is going to cause an epigenetic shift in the expression of specific genes. Okay, now here's something that I guys want you to see. Okay, in regards to concerns about inheritance, when an organism's body cells divide in mitosis, the new daughter cells can inherit the same epigenetic markers as the original cells they came from. This inheritance can be passed from cell to cell, as we just discussed earlier. Now, when a sperm cell and an egg cell combine to form a fertilized egg, which occurs in humans and many other organisms, epigenetic markers are often cleared. However, some epigenetic markers are not cleared and can therefore be inherited from one parent to offspring in this way. Now, that's something very important to take in mind, you see? Now, let's go ahead and talk about vitamin D mediated epigenetic regulation. This is very important, okay? So as we all know, vitamin D comes from the sun. It's thanks to the presence of UVB rays. This is going to coincide on top of a type of cholesterol known as 7-dehydrocholesterol and our skin, and that's going to be transformed into cholecalciferol. This eventually, cholecalciferol is going to bind to a protein known as vitamin D binding protein, and it's going to take it to the liver. And over here, we're going to have a cofactor known as magnesium, which is going to participate in the hydroxylation process of cholecalciferol, thanks to an enzyme known as 25-hydroxylase, and transform that into calcifidiol 25-hydroxyvitamin D3, right? Now, this goes all the way to the kidneys, and it's also transported thanks to the vitamin D binding protein and thanks to an enzyme known as 1-alpha 25-hydroxylase in the presence of magnesium as a cofactor. Once again, let's not forget that magnesium participates in more than 300 different biochemical reactions in the body, right? We end up making calcitrol, also known as 1,25-dehydroxyvitamin D3, which is the actual hormone, the actual vitamin hormone D3. This metabolite, which is the most important one, is the one that's going to be distributed throughout the body. But bear in mind that this metabolite is also considered as our reservoir of vitamin D. It's a prehormone known as calcifidiol and has a half-life of approximately 2 to 3 weeks. And this calcitrol, which is the actual hormone, has a half-life of approximately 2 to 4 hours. So it's very short. This eventually is going to act upon the vitamin D receptor that's found basically in every single cell in our body except red blood cells. Okay, and one of the things that it's going to do is that it's going to go all the way into the cell. Here we have a cell wall. This is the nucleus of the cell, and that's where we find our vitamin D receptor. And it's going to go ahead and bind to this receptor, all right? And once it binds to the receptor, a process known as heterodimerization takes place, which changes the configuration of this VDR receptor and allows for the retinoid X receptor to bind to it, okay? Now here, this retinoid X receptor, we have to remember that it's activated thanks to retinoic acid. And retinoic acid derives from vitamin A. So vitamin A is very important as a cofactor, right, to be able to form what we know as the vitamin D receptor complex, or also known VDR-RXR complex. All right. So this is an actual picture of how it actually looks like. You see, it's very complex. Anyways, but we also have what we call the Krebs binding protein, and also another protein known as P300. And this binds also to the VDR complex, activating a series of things, and these are known as coactivators. And these coactivators possess histone acetyltransferases, right? HAT activity, promoting histone acetylation and chromatin remodeling to facilitate gene expression. Remember that image I just showed you guys? Well, there you go. It's facilitating gene expression. So these guys are coactivators that facilitate gene expression. Now, here we have these other two, known as NCOR and SMRT. These are called co-repressors, right? Now, co-repressors mediate histone deacetylation and chromatin condensation, leading to gene repression. So these silence genes, all right? And there's a very nice balance between all of these together. And when we have all of this set up nicely and all of these cofactors are working all together, the coactivators, the co-repressors, the retinoid X receptor, the vitamin D receptor, the metabolite, all of these make up what we know as the vitamin D receptor RXR complex. Now, here we have our VDR complex, and it's going to go ahead and it's going to bind to what we call vitamin D response elements. Here we have our genetic DNA, right, our genetic code, and here we have a sequence of genes, right? Here we have a specific target gene, and at this specific target gene, we're going to have what we call vitamin D response elements. And here, the VDR binds to these VDREs and activate what we know is the target gene. So this binding initiates a series of molecular events that regulate gene transcription and ultimately influencing various cellular processes, including the absorption of calcium, bone mineralization, immune function, cell proliferation, and differentiation. Okay? Now, don't forget that this is all happening at the level of the nucleus, okay? And there's a feedback regulation here where it says the activity of VDR complex is tightly regulated by various feedback mechanisms, including the availability of vitamin D and calcium, as well as the expression of levels of cofactors and target genes, as seen over here. See, this ensures precise control of vitamin D mediated signaling pathways and cellular responses. Okay, so let's say that we have here a patient who has psoriasis, right? Okay, as we know, here's a target gene. This target gene, known as IL-17A, plays a crucial role in the initiation and propagation of inflammatory responses, all right, particularly at mucosal surfaces and barrier tissues, and also in the skin as well. While interleukin-17A is a mediated inflammation is essential for the host defense against pathogen, when you have a dysregulated gene, interleukin-17A signaling, means that pathogenesis now can take place, leading to inflammatory responses and also the presence of autoimmune disease such as psoriasis, as we can see right here in this picture, right? Or conditions such as rheumatoid arthritis, inflammatory bowel disease, and asthma, etc., right? But when we have our VDR complex here and it binds to this VDREs, it's going to sometimes silence down this gene and allowing for this interleukin-17A to stop being produced and stop initiating the propagation of an inflammatory response and allowing for this patient to be able to heal, as you can see right there, right? So you can see this is very important. Now, as I did mention, the microbiota has many different functions. One of them is digestion of fiber, vitamins, neurotransmitter synthesis, hormones. It also educates the immune system. Let us not forget that digestion of fiber is important for making acetate, propionate, and butyrate. Now, look how cool this is. Small short-chain fatty acids or small-chain fatty acids produced by the gut microbiota are also known to inhibit and promote histone PTMs. All right, these are enzymes. Butyrate and propionate are inhibitors of sirtuins deacetylase enzymes. So these are enzymes that are going to participate in the deacetylation. Now, acetate from the gut fermentation contributes to the pool of intermediate molecules known to form acetyl coenzyme A. This is a precursor for the Krebs cycle, a major acetyl group donor, and also helps in histone acetyl transferases. Okay, very important. Look at that. That's pretty awesome, huh? And finally, here we have acetate is also known to be an inhibitor of histone deacetylases, increasing histone acetylation levels and regulating chromatin accessibility. So now we can see that these small chain fatty acids or short chain fatty acids are now making all these enzymes that are responsible for deacetylation or histone acetyltransferases or histone deacetylases. Boy, we have a lot of things going on at the same time, and thanks to these three major small chain fatty acids. Now, in regards to vitamins, we know that the most amount of our vitamins comes from our diet. But we also have to recognize that the bacteria that are found in our microbiome also have the capability of being able to make specific vitamins, such as, for instance, Bifidobacterium species are able to produce folate and also vitamin B12, right? Folate is a vitamin B9, which is very important for the folate cycle, and also for the methylation cycle, right? These both are together. Both of these work together. Then we have lactobacillus species that also make vitamin K2. Then we have Bacteroides species that make vitamin B7, vitamin K1. Then we have Escherichia coli that's responsible for making vitamin B12, Propionibacterium species that make vitamin B12. So as you can see, many bacteria that are found in our microbiome are also capable of making vitamins that will contribute to these metabolic processes in regards to the methylation cycle and also cofactors that are responsible for epigenetic expression. Okay, so most vitamins and minerals come from our diet, but the microbiome contributes more when there is a lack or a need of them. Okay, as you can see, the microbiome is working always in trying to find a state of homeostasis, as you can see. Now, this is an excellent slide. You can find this at this article right over here. Okay, here we have our gastrointestinal tract. As you can see, we know that a lot of these vitamins, such as folate, B6, B2, B12, all come from our diet, and they're going where? They're going to be acting upon the folate cycle over here, and also the methionine cycle over here, being able to make methyl groups that are going to be able to go silence specific histones and DNA markers, and But if you see over here, we also are making short chain fatty acids such as acetate, and acetate is a precursor molecule for the making of acetyl coenzyme A, which is going to be a molecule that is essential for the TCA cycle, or also known as the Krebs cycle. You see that? And alpha ketoglutarate is also very important for the regulation of all of these histones, because we also can add methyl groups over here. And then we have butyrate and propionate that are also going to be acting on so many different enzymes that are going to either participate in the acetylation or the deacetylation or the methylation or demethylation of our genes. So we can definitely now say that short chain fatty acids such as acetate, butyrate, propionate do contribute in many ways to the way how the TCA cycle participates, and at the level of the mitochondria, or how the TCA cycle or the Krebs cycle is going to be able to work correctly under the presence of adequate amounts of acetyl coenzyme A, because acetate, which is a small chain fatty acid, comes from our microbiota. So you can see the microbiome has a very big impact on metabolism and human metabolism in many ways. Well, I hope you guys enjoyed this module. My name is Dr. Eduardo Beltran, and I'll see you next time.